At least I can see it. Yes, please go ahead. Yeah.
It is useful. Thank you, Dr. Srinath, for helping me out. So, if first slide is available, then I can start. Okay. Good afternoon to MycoIndia for giving me the opportunity for this presentation. And I hope this presentation provide you some insight how fungus are useful in bioremediation purpose. Because I'm basically an environmental scientist and using fungus for the agents of remediation and got success in some of the cases. So my presentation includes 4 different avenues where I have used fungus in our research. So we have the next slide, Dr. Shina. Okay, first is regarding the arsenic problem, because arsenic is one of the potential carcinogen as per the WHO. It is having the least limit of 10 ppb per liter for the consumption, and it is being accumulated in the food chain, and paddy is the most susceptible crop. So with some DBT-sponsored project, we carried out this study, and we have developed one synergistic fungal formulation of some of the novel strains that we have enumerated from the paddy fields contaminated with the arsenic, specifically in the West Bengal region. Next slide, please. So, if just giving the background of this arsenic infestation in this country, the 12 states are heavily contaminated with arsenic in potable water as well as in the groundwater system. Next slide, please. So, Central Groundwater Board of this country and Ministry of Agriculture, they have already mapped 9 states and 10 states respectively, and about hundreds of districts are having the contamination more than the threshold given by both in groundwater contamination condition. For potability, it is 10 ppm, as I told earlier, and for contamination threshold provided by the FAO and European Union, it is 20 ppm. So this much districts is being on the threat of arsenic contamination in the food chain. Next slide, please. So coming to the health hazard, it's causing the several neurological, dermatological, gastrointestinal, cardiovascular, and carcinogenic disease. And earlier it was the MCL was 50 ppb, which has been reduced to 10 ppb. And you name the crop, if the irrigation water is having the arsenic contamination, it is being accumulated in the food crop or the vegetable or even in the fruit. Next slide, please. These are some skin element, the real-time picture which we have taken from the Jadavpur University, and it's a very, very serious issue in the Eastern UP, the West Bengal, the Jharkhand area, and Assam also. Next, please. So to ensure the safe level of inorganic arsenic, so this modus operandi is starting from the monitoring and assessment of arsenic level in soil and water, then some water management issues, the aerobic rise concept is there, rhizospheric manipulation with nutrients and microbes. So we have chosen this aspect of having the safe level of inorganic arsenic in the rice, and which would be the safer to work. Next slide, please. So started with the sampling of paddy soil in the contaminated region of Uttar Pradesh and West Bengal. We got some culturable strains, then we did the tolerance study and removal assay at lab scale, and that has ended up in with elucidation of 4 fungal strains as a best performer out of 69 culturable strains that we got for the arsenic remediation and plant growth promotion ability. And first time we have reported this arsenite methyltransferase gene in our one of the potential fungus strain, West Radicula aurantiaca, and this is the first report across the globe having the eukaryotic origin of this detoxification gene, arsenite methyltransferase. Next, please. Then we started developing the bioaugmented based safe cultivation practice to remedy the arsenic contamination to the crop. And we have developed the fungal consortia and did the compatibility assay and carrier screening. And we have purposely selected the arsenic prone site in West Bengal, namely the Getugashan Gotra of Chagda block and Pipligram of Gayagata block, to conduct the field trials. Next, please. And we have chosen the three kind of exposure of our fungal consortia in a form of seed soak, root soak, and the soil application. And we have screened the bororized germplasm of paddy variety of West Bengal being cultivated by the farmers over there. And the 4 arsenic accumulating rice varieties, namely the IR36, Chitesh, Lalat, and Trugna, we have selected to carry out this split plot experiment on those 3 sites. In 4 consecutive rice cropping season, the Boro and Aman, Boro season, they have named after because they are totally depending upon the borewell for irrigating the rice cultivation. Next, please. And with these, we found that about 50% reduction in grain arsenic content and soil bioavailable arsenic content reduction of about 40% that we have achieved. And with that to check our fungal strains were thriving in the rhizosphere. So we took off the rhizospheric sample for estimating this soil cholesterol, which is the only live biomarker of live fungal cells. And we got the success up till now. And now we are doing further research. Next, please. So now we are attempting the genetic improvement of novel fungal strains because our fungal strains individually possessing the different arsenic detoxification traits like volatilization, oxidation, and sequestration. So 2 approaches. One is the RSI gene knockout, and the protocolus fusion is the second method we are applying. Next slide. So coming to the whole genome study of this arsenite methyl fungus, Vestridicella orangeaca, we have completed. And one of the curated arsenic database which is freely available to us, and we have mapped our whole genome data to this curated arsenic database. Next, please. So this gene is of our interest because this fungal strain was reported to have about 60 to 65% of enriched broth culture volatilization of arsenic content. So we intend to silence this gene of arsenite demethylase, which is a non-heme ferrous iron dependent oxygenases, and this is catalyzing the oxygen dependent cleavage of carbon dioxide arsenic bond in the that is present in the trivalent organoarsenicals and just converting them into the inorganic arsenic. So this is a barrier to get more volatilization by this strain. So our idea is to knock out this particular gene by using the CRISPR approach. So we did the blast with the whole genome assembly, but we couldn't find the significant matches. Again, we thought of considering this VOC domain, which is occupying a major portion of the gene, and we successfully find 6 sequences of this VOC domain, and now we are doing the RT-PCR to find the putative demethylation in these 6 sequences. Next, please. So these are the 6 sequences that we found in whole genome assembly, and we are keeping our fingers crossed to get some success. Next, please. This is the age-old technique of protoblast fusion, and as I told all of you earlier, that we have the distinct arsenic detoxification feature in our individual fungal strains. So we, with the PEG, we have successfully find this fusant, and with fusant regeneration we have attempted. Now we are in the process of stability assay, and just to see the arsenic methylation in the fusant, what are the biochemistry involved in that. So the analysis is going on along with the confirmation of genes exist in the fusant. So these two approaches for arsenic detoxification using the fungus we are trying. Next, please. So there's the second avenue. I hope all of you are knowing this SCH hexachlorocyclohexane residue in the soil. So we are attempting the mycoremediation of this reduction of SCH residues in the soil. Next slide, please. Giving you the overview of SCH is a polyhalogenated organic compound which is mostly used for the insecticidal property, and gamexine is the trade name of gamma isomers. So SCH exists in the 6 forms: alpha, beta, gamma, delta, and 2 others. But basically these 4 are existing in the SCH mud. And if we produce the SCH, per ton of SCH, the waste disposal which are carrying the alpha, beta, and gamma isomer of SCH, we are having more than 8 to 10 tons per ton of SCH, gamma SCH production. So it's a carcinogen, and mostly the bacteria, sphingomoniads, are reported for degradation of this SCH content in the soil. Next, please. So we carried out our studies at a dump site, which is 35 kilometers away from our institute at Lucknow, and we find that the rhizosphere zone of some plants which are growing over there having the microbes which may have the ability to degrade the different isomers of SCH to the enzymatic action. So just to begin with this idea, we have isolated some fungal strain. Yeah, please, next slide, please. Earlier ones. So okay, this about the publication of that finding. Next slide, please. So with the enrichment culture of about the 100 ppm of SH isomers, that has yielded the 9 potential fungal strains, and we have carried out the different biochemistry, biochemical assays like the dechlorogenase assay, then the linden degradation, how much linden was there left over in the broth as well. So we found that out of 9, the chlorostoma is the most efficient, most potent one. And regarding the degradation at the genomic level, the lin-A is the responsible gene. So we did the confirmatory match of lin-A in this chlorostoma richardsii. Next, please. So this was the pathway suggested in the sphingomoniads, starting from the lin-A, and this is the upstream pathway that is involving the lin-A and lin-B. Then 9 others lin pathway enzymes existing in the downstream pathway. So next slide, please. So we analyze the what kind of intermediates are being formed by the chlorostoma in our broth in this with the SH gamma isomer, and we found that the upper stream intermediate metabolites which have been reported in the case of sphingomoniads is also being reported in our case. So there is a confirmatory study of linden degradation using the lin enzyme in our chlorostoma. Next, please. This is study also being recently published in the BRT. Next, please. So there's the last one. The cyanide is another constraint in the industrial wastewater, mostly of the steel industries. Next, please. Giving the overview of cyanide, it is being formed in the coke making process, and which is a common issue across the world in the steel industries, and volume of effluent having the loads of cyanide ranging from 2 to 20 ppm is very high. It's about the 100 cubic meter per hour of effluent is to be treated. And mostly these are the two processes in the steel industry. One is the blast furnace, and second is the coke oven wastewater, which is having the cyanide more than the threshold provided by the Central Pollution Control Board of this country, which is the 0.2 ppm in the discharge. So there are the different pathways of microbial degradation of cyanide. One is the hydrolytic, reductive, oxidative, and substitution reaction. And we are focusing on two aspects. Second is the hydrolytic, and first for which we got some success at the lab level is the Rodinase enzyme, is the substitution reaction which convert free cyanide into the thiocyanate and reducing its toxicity. Next, please, sir. So we have taken the samples from the steel plant at Jamshedpur, and we have isolated some bacteria and fungus. Trichoderma strains, we got the maximum degradation of free cyanide content in our lab assay. Next, please. And we found that it's a first order free cyanide removal kinetics that being observed in our trichoderma, two of our trichoderma strain. One is the Lambda-Bacterium and second is the Staphylosporum. Next, please. And secondly, as we move down, then we have immobilized these two culture of trichoderma strains in the immobilized form using the alginate bead, and we found that within the 72 hours, these consortia of these two fungus in the immobilized form able to decrease the free cyanide content up to the 99% level, and we are upscaling this process. Next, please. And we have also published this in the BRT. So next, I think this is the last one. So for me, the mycoremediation is a win-win situation, and I just showcase you whether it is the iron or the metalloid or the organic content, organic contaminant. Our fungus beautifully is are having the pathways, the biochemical pathway. You can stop sharing the presentation, sir. Thank you, Dr. Shinai, for giving me this opportunity to interact with the mycologists of this country and the young fellows. And definitely, the fungal are the super agents provided by the nature. And for remediation of organic as well as the inorganic contaminant, we are utilizing these resources. Thank you so much. Thank you all.
Thank you very much, Dr. Srivastava ji. I mean, the Western Dicala strain is quite
because at the time Dr. Chinai was at MTech, and he helped us, and we happily shared one paper. I think the first paper you
yes.
were the co-author. I'm thankful to you for that for that help.
Thank you very much. We'll take a couple of questions, maybe more. Mr. Arvind. Wait, let me just help them turn on the mute. Okay, Mr. Arvind, your turn.
Yes, sir. Thank you for the opportunity again, sir. So, as you mentioned, the arsenic reduction will be around 50% when it's gone with the fungus. So, in case during the first cultivation period, there's a 50%. If there is again a second cultivation or the continuous cultivation is being occurred and using the fungus again and again, so how is the probability of reduction of arsenic in that field? So, just curious to know.
If you recall that slide, so initially after completing the first bororized experiment, the success rate was only 24 to 25%. And we have conducted a subsequent trial at the same field with the same split product, and that has increased from 24% up to the 50%.
Wow, that's great. So, okay.
And that is the maximum one.
Okay, fine. So, in that cases, the accumulation of arsenic does have any changes on the fungus in morphology or in the activity, anything? It's been showcased, so?
Because it was difficult to reculture those fungal strain, and now we are tagging those fungal strain for this kind of study, because it is very easy to conduct this kind of experiment here at your lab scale, but in the field. So what we did this live biomarker study of soil, rhizospheric soil ergosterol content. So as of now, I have the result of those only, but it's a good suggestion, and we are planning to have those data set with us.
Okay, so then again the final query, as the accumulation of the arsenine occurs in the fungus, does any fungus do any modification for the oxinate for being less toxic to its survival? Because when you say it's for the 25% to 50% increase is there, so means the accumulation process being continuous for its lifetime of the survivability. So there's any modification happening, or the fungus is reacting towards the oxinate? There's anything, sir?
If you consider that the fungus is adopting the multifarious detoxification mechanism. First is the conversion of trivalent form into the pentavalent form, and it is again solved at the mineral surface. Second is the volatilization, and once the third methyl group is being attached with the help of methyl transferase, it gets volatilized into the less toxic form of arsine. So these. And third one is the sequestration, vacuolar sequestration of this PC conjugated, phytogelatin conjugated, and this arsenic derivative into the vacuole. So these three detoxification mechanisms being facilitated by the fungus.
Okay, thank you, sir. Thank you for the insight, sir.
Thank you.
Pavitra, please ask your question. Thanks. Pavitra? Okay, Aarti? Okay, Nita Fatima. I think we will let you go now, Dr. Srivastava. I think we'll continue with the next presentation. Thank you very much, Dr.
It's my pleasure. It's my pleasure, and
thank you very much. And it's good to know all the work you have been doing, fantastic work you have been doing at NBRI. Thank you very much, and we hope to see you once again at MycoIndia in coming years or coming months.
Definitely.
Thank you so much.
Thank you.
Thank you. Now I request Dr. Poonam Mishra to give her a presentation. Dr. Poonam, can you hear us? Dr. Poonam Mishra, can you hear me?
Yes, sir, I can hear you.
Great.
Am I audible to you?
Yes, you are. You are audible. Thank you very much for agreeing to give this talk on fungal plant pathogens. I request Dr. Gunjan Sharma to introduce our friend Dr. Poonam Mishra to today's audience. Dr. Gunjan, please.